All right. So today I'd like to introduce our speaker. Dr. Alex Petman is an assistant specialist in cooperative extension and also an assistant plant pathologist. Today he's going to be speaking on downy mildew of vegetables. And now, Alex, I'll pass this over to you so you can go ahead and share your slides.
Okay, so yeah, good afternoon, everyone. Thank you all for being here, as joining virtually. All right, so let's get right into it. So first, we're going to talk about just as a little outline slide here. So we talk about downy mildew pathogens, a little bit about their host range, and then a little bit about the disease cycle with specifics about their sources of the pathogen. Okay, so downy mildew. So what are they? They are water molds like Pythium or Phytophthora. It's a different group of organisms, so they're not true fungi. And so fungicides used for their control are typically specific to this group. They are what we like to call this fancy term obligate biotrophs. Which means they require a living host to grow and reproduce. They can survive in, outside of a living host, but to actually grow and produce spores and complete its life cycle, they need a living host. And there are a lot of them. One number we have here is over 700 different species, and most of them are generally host specific. And then, so what does that mean? So for example, if we have downy mildew on lettuce here, and this downy mildew Bremia lactucae will only infect lettuce. So if you take your lettuce with downy mildew and go rub it on a healthy onion plant, and you have, of course, favorable environmental conditions, that onion will not get a downy mildew from that lettuce plant, and vice versa. And the same goes true for spinach. Now within each, there's maybe some ability to cause disease on closely related plants. So, onion downy mildew is primarily a problem on onion in the field, as we know, and it may be shown to infect other things like garlic in maybe more controlled conditions, but it's generally, garlic, downy mildew of garlic is not a huge problem in the field as far as we know. So, even to closely related plants, it's not a strong pathogen or a common pathogen of. And go and drill more in depth here, looking at some of the plants in the amaranth family. So, I had spinach on the previous slide, but let's look at some more closely related plants, like the, a goosefoot or lamb's quarters or quinoa. These are in the same subfamily within the amaranth family of spinach, but actually they're affected by a different species of downy mildew. So Steve Koike and Jim Carell, Steve Koike formerly of Cooperative Extension in Monterey, has done some of this work and shown that they've actually done these experiments to show that these are different. They do not cross-infect between spinach and goosefoot or lamb's quarters, those pathogens, and also this downy mildew Peronospora variabilis of quinoa and goosefoot and lamb's quarters, has been characterized in other studies related to quinoa. So also for beet and sugar beets, it's characterized as a different pathogen. This sugar beet is in a different subfamily within the amaranth family, and probably not going to have cross infection between spinach and sugar beet. I just have this probably not here because this particular experiment hasn't been done to try to infect beet or sugar beet with the spinach downy mildew Peronospora fusa, but we think it's very unlikely that we'll be able to infect sugar beet. Okay, and looking at a few others, just as a quick example, we also have a downy mildew of basil, or downy mildew of brassicas. These are affected by different species. For the brassicas, we know that these are, broccoli and cauliflower and sprouts. These are all cultivar groups, so they're all the same species of plant, and so the The literature is a bit kind of a little bit unclear, confusing on this, but I would suspect that the same pathogen is affecting all these different cultivar groups of of broccoli and cauliflower and things like that. For some things that are more distantly related, like some of these cabbages or other species, we're not exactly sure. And I do have this note here about Bremia lactucae. You have this question mark here because we don't think it's the same. We don't think it's the same pathogen population necessarily, but there can be a cross infection. So not a lot of information on this that I know of, but just heard some things that it may not be the entire same population that affects lettuce that is grown in commercial fields and the weedy prickly lettuce. Okay, so just looking at the basic disease cycle of lettuce downy mildew, we start with some spores, and they are, and this is the primary way that this pathogen, and this goes for other crops as well, a primary way of these airborne spores that they get around, so they can be blown by wind. And so they land on a healthy leaf and germinate and penetrate into the leaf. Then after a certain amount of time to incubate, the incubation period, some symptoms develop, and then at that time or shortly thereafter, a new crop of spores is produced. And so this time between infection and sporulation, what we call the latent period, can take 7 and 9 days for a lettuce downy mildew. And then following sporulation, those spores, this new crop of spores, can be blown to a new plant, new healthy tissue to begin another cycle of infection, which can happen as many times during the course of a season. Okay, so looking at some research we're doing about when these spores are present. So this may be good information to know if we want to protect our crop or know when the disease is at highest risk. And so this particular project, we are trapping spores for both spinach and lettuce downy mildew. And we're doing this in, at least in the desert areas, September through April, 2 to 3 times per week. And just showing an example of Coachella here, where we have these traps. They're kind of at the north and south end of the valleys. And this arrow indicating the primary direction of the prevailing winds in the kind of the winter season. So this project is led by Steve Klosterman, who's a USDA scientist in Salinas, and also includes Ali Matnazar, who is a farm advisor in Imperial. And so this is done in Coachella as well as the Imperial and Salinas valleys, and it's been funded by the Leafy Greens research program as well as CDFA. Okay, so looking right at the data here, this is for a lettuce downy mildew. So what we have here is it broken down by each valley, those 3 valleys I mentioned, and the data is summarized by on a weekly basis. So on a week ending that particular day, the data is included for the week ending on each Sunday, and each dot or square or triangle indicates the average level on that week, and the bars indicate the maximum and minimum levels of that week. So this is combined for those 2 locations and 3 times per week. And then we have the different shapes and colors indicated by kind of a indication of the risk, relative risk based on the numbers. And so kind of have these zones here where, even if we have some values here, we think it's probably a negative detection. And so what we're doing is we're detecting the DNA of the pathogen. We're kind of doing some extrapolation or estimation on the number of spores that means. So it's take this with a grain of salt. But basically, as we get higher, there's likely more spores and more of a risk in terms of disease. And also note this is on a logarithmic scale, so due to the numbers of spores, so it's not a linear scale here. So basically, in Salinas, we were seeing many more spores, and pretty consistently over the course of this trapping. It's by this measure, the spore pressure is consistently moderate to high, even during kind of the January, late December, January, February timeframe when there's not a whole lot of lettuce, and that includes there's this on the weekend in January 2nd, there's maybe a negative detection. That's a little bit after the free period in Monterey County. So, even during that time, when there's not a lot of crop in the ground, we're still seeing some detection there. Now, looking at Imperial, on the other hand, we're not seeing a lot of detection in September and October, and and this was true in Coachella as well. Not seeing that much detection until the end of kind of when those first plantings are to be harvested, and then and comparatively for comparing Imperial and Coachella, just not a lot of spores detected in Coachella, pretty inconsistent, whereas in Imperial, you have this obvious strong peak here that kind of equaled the levels that we saw in Salinas showing up in December and January. Okay, just checking the chat here. Okay. Okay, yeah, so that's
Alex, could you maybe clarify on this graph what the negative detection means?
Yes. So the way we detect this is through a procedure called a quantitative PCR or qPCR, and it's detecting DNA, and so it's not a hard and fast detection. So you basically wait to see, it goes through several cycles, and the cycle in which you first see a signal is kind of your detection level. And basically this thing can, you can still get a signal really late in the cycle, but it's kind of a determination. You have to make a judgment call, like what is the cycle you want to say, oh, this is a positive and negative detection. Because if it keeps cycling, it can kind of, it can get some false positives or some ghost signals. And so you have to make a determination. And so this is basically, even though there's a data point there, we're saying it's a negative detection. It's meaning it's just, there's just not something really there.
Thank you.
But that's a good question. Thanks. Okay, so now looking at spinach, it's a little different. We don't have, they were able to do these levels of risk based on some field work that I'm not gonna, don't exactly remember the details, but they have a little bit of better handle. They did some field studies where they correlated the levels of spores to what actually happened in the field, but they don't have that for spinach, so we don't have those levels of risk. And so we're looking at kind of a less certain classification of these different levels, but still but we can still look at these and say, at a certain level, we have a really, pretty strong detection that we're confident is a true detection. And we can see here the pattern is pretty similar to lettuce. We have, semi-consistent detection in Salinas with a big peak there in October and November. In Imperial and Coachella, little to no detections in September, October, and then you get a spike beginning in November and kind of, at least in Imperial, continuing to some degree through that winter season. Whereas in Coachella, even we're a little unsure how much we really detected there. Maybe only a few were in the maybe range. but yeah, overall pretty similar to lettuce. And so just looking at these together and putting them in the same kind of categorization as both the lettuce and spinach, lettuce on top and spinach on bottom, just for comparison purposes, to summarize, in the desert, we didn't detect significant numbers before we had mature crops or before there was significant acreage. And of course, when things started to cool down, which we'll talk about a little later. And then so kind of the early conclusion, tentative conclusion we can draw from this is that this suggests the spores were not being carried long distance from another region. Because if you can imagine, we have this spike here in Salinas, in October and November. So if those spores were being blown all the way from Salinas, we maybe should have picked up a little more. Or at least blown in significant amounts. We maybe should have picked that up a little bit more in the desert in September and October. Or so it's at least, it's not being transported long distances in a large number or enough to detect. And so, that could suggest it's kind of a, more localized or regional sources of airborne spores is more likely. Okay, so looking a little bit more in depth onto the disease cycle of lettuce, for example, here. So we talked about this in-season, what we call polycyclic cycle that can happen many times of spore production. But in between seasons, there's another part of the disease cycle of survival. And so this is maybe more relevant to places or regions that experience frost or cold winters. And so, but this is kind of a general part of, a general downy mildew cycle. It could apply to downy mildew of various crops or different species of the downy mildew pathogen. And so the idea here is that you have some symptoms, but instead of going to a sporulation, what can happen is that it produces these oospores, which are the thick-walled, you can see, double thick wall spores that are adapted for surviving unfavorable conditions, like a hard winter or just unfavorable conditions. And so this I write suspected here because I'm not sure if this has been proven for the lettuce downy mildew or Borrelia lactucae, but this is kind of a inferred or suspected part of the disease cycle for many downy mildews. Okay, and then let's look at all those. We talked about these spores, and so and then we said, oh, maybe they're airborne, within a region or within a season. But now we want to think, well, where does the spores come for that first infection? They can't just be flying in the air forever, right? So now let's look at potential sources of the pathogen for that first infection. So say, there's no spores coming in from outside the region, and there's no disease, say, in the Imperial Valley. We're gone through the summer, and so we're starting into the winter season. And so where's that first infection going to come from? And so, well, they could, if you have your fields, they could come from somewhere outside the field, and they could, of course, airborne spores, as I mentioned, is the primary way the pathogen gets around. So they could come from nearby an adjacent field, or they can come in smaller numbers from a field farther away. And this is this movement has been documented for other downy mildew pathogens like cucurbit downy mildew on the East Coast. They track it through the season as it comes from overwintering regions in Florida, and it gets kind of blown northward progressively through the entire year. And so it does move by wind, but not super long distances as far as we know. But yeah, so that they could come from different distances outside a given field. But as we look at maybe where could it come from that initial infection, or some other possible sources? It could come from in the form of previously infested residue. Maybe it was a diseased plant that that survived over the summer or winter, depending on where you are, and it's maybe the tissue didn't degrade, it wasn't broken up during cultivation or something like that, and that can survive between periods when there's no crop. Another similar kind of source is a volunteer plant, so maybe the uncontrolled a volunteer plant, or kind of sprouts back up and gets infected and survives for the next crop. Another possible, as we talked about, was those oospores that can survive in soil, and this could also happen in the field, and this could come from crop residue that is infested at the end of the season. The oospores, that material could fall off the plant and get incorporated into the soil, and then oospores could be produced in that material and then survive into the next season. And then another possibility, if there's no pathogen present in that field or in that region, could be seed. And this is what we talk about, one example, but has been found for many downy mildews. It can survive in the seed as oospores. And this could allow a movement across regions of the pathogen. And that kind of have this asterisk as theoretical. This is kind of a general disease cycle. Just thinking of from a plant pathologist perspective, all these possible sources. I have pictures of onion here, but that doesn't necessarily mean these things are definitive. But kind of just using it as an example to show some pictures of diseased plants. Okay, now let's talk about a little bit more specifics. In this case of onion downy mildew Peronospora destructor, what could be those potential sources for first infection? If we say, oh, there's not going to be no infection in the region, so there can't be any airborne spores, or what could happen in that situation? So it could happen, in or could be present in or next to the field. And one example is that soil that I've mentioned. And so this group in Canada did study where they were able to use a molecular method to detect the pathogen in the soil, and they were able to do that. And they were able to find it in a lot of soil samples. And so, they grow onion pretty commonly up there, and they have rotations of different lengths here. So what you're seeing here is that they were able to find out the last time that this field had onion from where their samples came. And they were able to show that the level of the onion downy mildew DNA that was in the soil was related to how long it's been since onion was in there. And so this suggests that this pathogen that they were finding in the soil was from a previous crop that had downy mildew, and then it was incorporated to the soil, and the pathogen was surviving there. And so so this is possible, this particular pathogen, at least in that climate. We don't know if this can happen in the in between crop seasons here in California, for example, in the summer in the low desert of California. Okay. Okay, now looking at that example of Peronospora fusa, a spinach downy mildew, Steve Klosterman's group at USDA in Salinas has been doing this work for several years now, and been consistently being able to find oospores of the pathogen in seed. And so they have this number here of, they've tested about 300 seed lots, and they found oospores in 50 of them. So about, what is that, 16% or so. And these spores are viable. So they've been testing it for 8 years, and they've found viable spores. And they've also demonstrated that oospores can germinate and infect roots. So this could be a potential how the pathogen would be surviving in the soil and then cause disease on a new crop of spinach. But I have this asterisk here. What hasn't been proven is that this is really happening in the field necessarily. So they're doing the experiments where they try to prove that this can lead to an epidemic, but they haven't actually definitively proven that. But it is, I think it is possible, and that's something that we have to consider when thinking about this. And for spinach, because we probably think this will happen at a low, very low frequency, that you'll have a plant in a crop has been infected by oospores, if it's happening at all. Okay, so now we'll move into epidemiology, weather and environmental conditions, and a little bit on management. Okay, so this is the disease triangle. It's a core concept in plant pathology. I'd just like to give a brief reminder is that a plant disease is an interaction, a abnormal functioning or unhealthy plant that results from an interaction between a susceptible host, a virulent pathogen, and all happening in a favorable environmental conditions. So you have to have all three of these things happen at the same time to get a disease. Okay, so looking at this for lettuce, and so we have a few, here we're looking at the favorable conditions for lettuce, and these numbers are reflective in the canopy. So if you have a thermometer and you stick it in the canopy, this is the values we're looking at. And so kind of broken up into two different parts to reflect two different stages of the disease cycle. So you have the infection stage, which happens in the morning, and sporulation, which happens at night. And so for infection, the primary thing that is required is a leaf wetness. So this is, literally drops of water sitting on the leaf. And for infection, there needs to be at least 3 hours of continuous leaf wetness for infection to happen. And this usually happens at beginning at sunrise because the spores are commonly released at around sunrise. And if the temperatures are above about 60 degrees Fahrenheit, this increases to about 4 hours. Now, of course, this can, there's always not a hard and fast rule, and you can the leaf wetness can vary throughout the field or within a plant, underneath in the nooks and crannies of a leaf. And so you can kind of just stand there on the field and say, oh, my lettuce is dry. But if you look closer, look underneath, the lowest leaves or in between, you can still get some leaf wetness that lasts longer. So be aware of that. And for sporulation, the big condition is having high relative humidity overnight or leaf wetness. And in both sporulation and infection, it requires cool air temperatures. And so for a lettuce, downy mildew specifically, they've worked it out between 40 and 70, with an optimum of 50 to 60 degrees Fahrenheit. And so these are things I think everyone generally knows. Downy mildews, lettuce specifically, as well as in general, are favored by cool and moist conditions. But these are some specific numbers that was worked out in a series of studies at UC Davis in the 90s. Okay, and now looking at onion, just had a very similar disease cycle in the season. Same as lettuce basically, but the latent period, that time between infection and sporulation, can be a bit longer, range from 8 all the way up to 16 days. So, if you go out there and see sporulation, depending on the conditions, the infection that led to that sporulation could have happened as much as over 2 weeks prior to that date. And now looking at the specifics on the favorable conditions for onion downy mildew. Again, this is in the canopy for conditions measured in the canopy. It's very similar, cool and moist conditions, but there's some specifics for onion, having no rain after midnight, and the previous day air temperature actually, the air temperature, the daytime before that night can affect sporulation, in which it's optimal under 80 degrees Fahrenheit, but disease can occur above that, especially because conditions, of course, in the canopy are different, generally cooler and can be more humid than the free air above the canopy. And then for infection, again, in the morning, having leaf wetness beginning at sunrise, and how much or the length of leaf wetness that is needed is proportional to air temperature. So under less than 70 degrees is optimal, which requires 2 to 3 hours of leaf wetness. Whereas if it's the temperature is warmer during those morning periods, a longer period of leaf wetness is needed. Okay, so I want to talk about our research project we have in this area and see if we can use these weather models as a management aid. And so just to note that these models were worked out in studies in Canada as well as in a couple places in Europe. So these haven't been tested previously in California. And so if we're looking at, if we want to use the environmental conditions as a tool to help us decide or think about what the disease risk is, there are a couple different situations we can find ourselves in. If the conditions are favorable for disease, if we're looking at the conditions in the canopy and we say, oh, it's favorable for disease. So then we can maybe take some action with that knowledge. We can maybe make a fungicide application. Maybe we weren't already planning on it, and then so we go out there and make it. Or we were planning on maybe spraying something, a low efficacy protectant fungicide, but now we know that it's favorable. Maybe we can switch to a more efficacious or more expensive product. The other situation is that if we find that conditions are not favorable disease, maybe we can say, oh, I don't need to make that fungicide application I had planned for tomorrow, or it's okay if it's delayed, or maybe we can switch to a cheaper product. And just a note about these particular models, what they're doing is they're looking at conditions that have already happened. And so we call these warning models. And so this is if you were to looking at a weather report on TV and they said, oh, the high temperature was, X yesterday, and then this is what we're looking at. We're not looking at the forecast and say, oh, the disease forecast tomorrow is A, B, and C, and so therefore it's favorable by this particular model using this data. Now, we all do this ourselves. We can say, oh, there's a storm in the forecast the day after tomorrow. It looks like it's going to cool down and maybe a chance of rain. So then that's more favorable for downy mildew. And so I'm going to take action. But in terms of this study, what we're looking at is weather observations that have already been made. And so for this particular trial in onion, we're monitoring conditions in the canopy. And so we have these leaf wetness sensors at an angle to mimic an onion leaf. And we're also measuring temperature and relative humidity in the canopy. And so this is data from a location at Brawley in the 2019 to 2020 season. And so this is showing air temperature, relative humidity, rain, and leaf wetness. And so for temperature, I've marked out the 80-degree threshold that I mentioned in terms of the high temperature, above which the production of new spores is slowed down. And then also that 95% threshold or 94 or 95% threshold for relative humidity, which is required for production of spores overnight. And so basically we can see, for relative humidity, it only exceeded that threshold a couple of times, and almost every time it was associated with a rain event. Whereas high temperatures kind of, they dip down below that threshold and were kind of cool, for many portions of this study, but did dip above it. And then as we started getting into April, May this is why it's cut off, because it's going off the chart here. And then we did have this major rain event here in the middle of March. Okay, now looking at the output from these disease models here, there's one main disease model called Downcast. There's a couple of variants of that one. There's a couple other models called MillionCast and UniMill, and then there's a standard calendar treatment. And so in this study, we were basically trying to time the fungicide application. So if an individual model was triggered and was said, oh, it's favorable conditions, then they would make the application. Whereas if I was in a standard calendar schedule, that's based on a kind of 7 or 14 week interval that is consistent and followed throughout the study. And so looking at these output here, what we have here is downy mildew favorability if we have a data point here that indicates the model was triggered, and the y-axis indicates if it's more or less favorable, because some of the models give you that degree. So basically what we're seeing here is we have the Brawley on the left and Hopeville location on the right. This trial was replicated at these two locations. See this big trigger event here in that early to mid-March that happened at both locations that coincided with that rain event I mentioned. And so at the Brawley location, we were able to make the application here, the first application of some of these models, whereas we had made an application in late February at both locations for the standard calendar treatment. And so this event happened to coincide with when everything started with COVID, and so we weren't able to travel and really make the applications that we wanted to. So that's why we don't have further applications or letters at the Hopeville location, because there was also some rain that prevented us getting in the fields at the Hopeville location. The roads were too muddy over there. But anyway, so we were able to make some of these applications, and looking at the disease, we found that these applications did provide disease control. And so here at Brawley, we have here the standard calendar treatment, which received Ridamil in late February, and then Dithane in mid-March. Whereas some of these model treatments only received Ridamil, but they attained equivalent levels of control here. Whereas some of these, we had a bunch of non-treated treatments, as well as one model that wasn't operating, so it wasn't sprayed. And so we had some more disease in these plots. And the disease were pretty low levels. These are really small numbers in terms of the symptomatic leaves per plant. So, not a whole lot of disease at this location, but the control level was clear here. At Hopeville, we saw a little bit more disease, and by the time we rated it, the tops had started to come down, so there was a little bit more difficult to rate, but we had clear control in the standard calendar, which received that Ridamil application in late February, whereas all these untreated plants had more disease, even though there wasn't clear statistical separation. And so as part of this project, we have this alert system in which the weather stations in the field send the data to a server, and then they compute the models and then spit out the results. And so we're able to put this in email form and send it to whoever is interested. In this particular case, we're just sending it to the people that have the weather stations in their field, and as well as for our research use to conduct the study. So we have also collaborated in Colusa in a seed field up there, as well as we have these locations in Imperial at our small plot trials, where we have these emails, and they're sent out on a daily basis. So this is potentially something we're interested in expanding. Just real quickly through this, we do have some times here. This is basically showing the number of times in the season that the model was alerted. And we have here several years and locations where we conducted these trials. And several of those years we had model alerts triggered, but only in the the 2020 locations of Brawley and Hopeville, did we actually see disease in the trial? So we had a lot of cases where the models indicated were triggered or there indicated favorable conditions, but we did not see disease. And so this goes back to what we talked about with the spore trapping on spinach and lettuce. It's valuable to know when the pathogen is present. So the situation here, why we didn't see disease, is because perhaps the pathogen itself was not present. Okay, so I'm gonna skip through the summary because I want to, we're getting a little behind schedule here, but basically we saw some ability of these models to time fungicide applications, and those fungicide applications made in advance of the disease did provide a good control under kind of low to moderate disease pressure. So let's move on to management here. So I want to try to pick up the pace a little bit. So talking about host resistance. And so just a reminder for those of you, for some of you, plant cultivars can possess some resistance gene that partially or completely prevent infection by a certain population of the pathogen. But through kind of the normal process, a new race can emerge of that pathogen that can cause disease on a previously resistant cultivar. And so for lettuce in particular, Richard Mitchelmore, UC Davis, has been doing this for a long time. And that group has found that at least as of 2019, these annual surveys find the majority of the samples have a novel or undefined pathotype. And for lettuce, there's about 9 races and kind of 2 variants, leaving a total of 11. But even then, they kind of find something that doesn't fit into one of those race categories the majority of the time. And it's a similar deal for spinach. There are now 19 races. The publication for the races 18 and 19 was put out last year, but they found it several years before that. And so, for example, Jim Carell at University of Arkansas does this work and found this this race with increasing frequency in the low deserts. They don't do as many samples as with lettuce, but there are some cultivars that maybe perhaps show resistance to at least race 18. Okay, moving on real quick to irrigation. And so the irrigation can affect disease susceptibility. And so we mentioned that leaf wetness is a big part of the disease cycle in terms of infection. And so Ali Montazar, farm advisor in Cooperative Extension Imperial, led this study looking at if drip irrigation is feasible for spinach production. And this was based on a grower who had been doing this for a few years, and so they wanted to look at if this is viable for wider use. And so we did the study in an organic field at the Desert Research and Extension Center in Hopeville, using a susceptible cultivar that's susceptible to all races. And this was field trials over 3 years. And so they looked at some few combinations of number of drip lines and kind of different nitrogen regimes as well. But what we're going to focus on is kind of just the drip irrigation versus sprinkler. And so here in 2020, we have this results for downy mildew. So here we have just percentage of plants with downy mildew. And we have a bunch of drip treatments at different nitrogen programs or different numbers of drip lines, 3 versus 4. But numerically, they all were lower than a sprinkler, and a few of them were significantly lower based on the mean separation letters above each bar in terms of statistical separation. And so the range of this difference was 2 to 7 times lower in drip versus sprinkler. And for 2021, kind of a similar trend. There wasn't as much statistical separation because the disease was not widely distributed in the field. There were a few hot spots in sprinkler plots that we should have a high level indicated here. And each point here is just kind of a different, had many different sampling points within a given plot. They were, these beds were a couple hundred feet long. And, this was true for different combinations of if it was either germinated with sprinkler or drip, and then followed with drip after establishment. But whenever it was under drip during establishment or after establishment, the disease was numerically lower. Okay, and so the primary mechanism that is likely here is that the sprinkler irrigation directly wets the leaves, whereas drip does not, and so this limits that leaf wetness that is essential for downy mildew infection. Okay, and then looking at fungicides, just a little quickly here. I think I may kind of skip through this table, but this is kind of a summary based on work that other people have done, including in Florida. And this is basically saying, looking at the number of trials that have been done, and if on the right here, and if the trial showed that this product or fungicide reduced downy mildew relative to the untreated check, then it shows up as a positive control. And this average percent of control is that magnitude of how much the disease was reduced. So for Arondis, for example, the average level of disease was 12% of that, or 1/8 of that's in the control check. And so I think this is kind of probably a review for a lot of people, but there's a kind of a groupings of how fungicides are effective, and with Arondis being very good, but only showing up in one, at least the data is only showing up in one trial here, whereas other things like Zampro, Arevis, and Reason are kind of commonly used for downy mildew control. Okay, just sorry for being behind schedule, but I'll just skip ahead a little bit. Wanted to talk a moment about fungicide mobility, and so this talks about where fungicides move after application. And so a protectant, it kind of dries right there. It does not move after drying, and so these are also known as contact fungicides. Some examples are mancozeb and copper and chlorothalonil. And the other group is penetrants, and so these are absorbed into the plant. And they can, different fungicides move different amounts within the plant once they get absorbed. And so you can have localized, which only move a short distance, translaminar, which means it moves to the other side of the leaf, or acropedal penetrants, which move upward only in the xylem. This is the water conducting tissue that only moves up, or a true systemic, which can move both up in the xylem as well as down with the phloem or the food or nutrient conducting tissue. And so there are only a few true systemics, and some examples in the context of downy mildews are the phosphonates, your Aliette or phosphorus acids, or Actigard. And so, as again, probably a review for many of you, but fungicides are most effective when applied before infection, regardless if they're of a protectant or penetrant. And while infections in an advanced stage cannot be stopped, some penetrant fungicides can slow infections that began before application, or have curative activity. But that window after infection can be narrow. So the commonly cited number is about 24 to 72 hours after infection. Was able to find a couple specific numbers. One in lettuce was kind of a small lab study, and they found that dimethomorph or Forum can have some post-infection activity up to 18 hours, but neither that or imidacloprid was effective at the next one they tested was 45 hours for lettuce downy mildew. So it wasn't effective up to that at that time. There is some data in hop downy mildew. They did this comprehensive study where they looked at a few active ingredients, and they found that kind of agreeing with that lettuce study that dimethomorph has some decent post-infection activity, but some of these others had kind of varying range up to a certain time frame. And this refers to the seeing a a decline in effectivity and efficacy relative to the preventative, after this time frame, they still have some level of control, but it's not the same level as you would get if you had applied it before infection. Okay, just wanted to wrap up that with about fungicide resistance, and that the continued use of pesticides could lead to fungicide resistance. And what it is basically naturally occurring mutation in the target site for the fungicide that kind of takes over the pathogen population as the fungicide is used. And this is the same principle that applies to the evolution of new pathogen races in terms of host resistance. Maybe I'll just mention a couple of risk factors. That's a single site mode of action, which a lot of these fungicides are. And when pathogen populations are high, so higher risk is happens when you have a curative application versus when the pathogen is not present yet in terms of preventative application. Or things like certain pathogens have a high evolutionary potential, like a lettuce downy mildew. And because it has a high potential, this pathogen has developed resistance to many fungicides, including erythrimyl or mefenoxam, the phosphonates, including Aliette. So resistance is commonly found in California. And one note for oxathiopyprolin, this is a very good product, and so we want to be judicious or careful about using it so we don't resistance doesn't develop, and we can continue to use that product. And just a note about onion downy mildew. Resistance has been reported in Australia and New Zealand. They've experienced poor performance for a long time, and at least in Australia. In Georgia, they've shown, or they've actually reported anecdotally, a decline in performance using Ridamil, but they haven't actually documented resistance. But as far as I know, we haven't actually documented resistance to Ridamil in onion downy mildew in the US, but it is something to be aware of, that it's happened internationally, and there are maybe some reports that it's not as effective in some areas. Okay, so that's it. I apologize for going over, but I wanted to plug a couple projects we're doing that we're looking for samples of disease. The first one is bacterial rots of onions. So we're looking at internal bulb rots caused by bacteria. If you see anything that looks like this, or you know what you're looking for and you have it, we appreciate you giving me a call. And we're also looking for samples of downy mildews of various crops like onion, lettuce, and spinach, as well as fusarium wilt of lettuce. So I think I'll leave this up for the moment, and I can probably, I don't know if we can start to look at the Q&A here.
Do you mind answering in writing in the Q&A? Because we don't have time to take them, the questions live.
Okay, sure, I can do that.
All right, thank you so much for staying on, answering all those questions.
Yeah, no problem.
And thank you for your presentation. So
yes.
We're going to go ahead and close out. Thanks again. And I hope everybody enjoys the rest of your day.
Yeah, thank you. Thank you all.